Every living thing on Earth, from the bacteria in the soil to the blue whale, is built from the exact same unit, the cell. Tracing these cells back reveals a critical divergence. This phylogenetic tree shows life splitting from a common root into two categories. At the top, prokaryotes, like bacteria and archaea. At the bottom, eukaryotes, like plants, animals, and fungi. This physical division dictates exactly how biological systems operate and store their genetic data. Before a scientist can align a sequence or query a genomic database, they have to master the structural differences between these two cell types. It is the mandatory starting point for any data analysis. This diagram of a eukaryotic cell shows its defining feature right in the center, a true membrane-bound nucleus. This nucleus acts as a secure vault. It houses the cell's genetic information, which is organized into multiple linear chromosomes. Prokaryotes completely lack this nuclear envelope. Instead, their genetic material, usually a single circular DNA molecule, floats freely in an open region called the nucleoid. These prokaryotic cells are incredibly small. A typical bacterium measures 1 to 10 micrometers in diameter. Eukaryotes, however, stretch from 10 to 100 micrometers. In 3 dimensions, this increase gives a eukaryotic cell a volume at least 1,000 times greater. Managing that immense physical space requires a completely different internal architecture to keep the cell alive and functioning efficiently. To handle their massive volume, eukaryotic cells rely on cytoplasmic organelles. Looking at this diagram, we see the folded structures of the endoplasmic reticulum and Golgi apparatus. These compartments localize specific metabolic activities. For example, this orange mitochondrion handles oxidative metabolism for energy. Prokaryotes operate entirely without these specialized enclosed organelles. All their metabolic processes happen in one shared internal space. This physical efficiency extends to their genetics. A prokaryotic genome is highly compact, dense with protein-coding genes, and relatively small. A eukaryotic genome is vastly larger. To fit roughly 2 meters of linear DNA into a microscopic nucleus, the cell tightly wraps its genetic material around specialized packaging proteins called histones. The genes themselves are also inherently complex. They contain large non-coding regions known as introns, which the cell has to carefully edit out through post-transcriptional splicing before it can build a protein. The segmented, specialized physical architecture of a eukaryotic cell is directly mirrored by the highly complex nature of its genetic code. When moving from viewing cells under a microscope to analyzing sequence data on a screen, these biological differences dictate everything. A massive linear genome filled with introns and alternative splicing events translates into enormous, highly intricate computational datasets. Querying a prokaryote like E. coli is a completely different process. Its high gene density and circular DNA require different mathematical alignment algorithms. Knowing whether a cell is prokaryotic or eukaryotic tells a bioinformatician exactly which specialized databases, sequence retrieval tools, and functional annotation programs. Ultimately, the physical anatomy of the living cell directly determines the precise computational tools required to successfully study its genome. The divide between prokaryote and eukaryote sits at the foundation of the tree of life, and it forms the structural basis for every dataset we use to decode it. To dive deeper into the physical boundaries that make these cells function, check out our next foundational video, Cell Membrane Explained. Make sure to subscribe to Bioinfo Bytes to continue mastering the intersection of biology and data science.